Hello, I'm Eric Archer, a former high school science teacher and current content strategy manager at Texas Instruments. Today I'm going to be going over the structure of DNA using the science-inspired lesson titled DNA Structure. This lesson and many more are available for free at www.scienceinspired.com. You can use these lessons with the TI-Nspire CX Graphing Calculator or the TI-Nspire CX Premium Teacher software. Okay, DNA, what is it? What's it made of? And why do we care? DNA is an acronym that stands for deoxyribonucleic acid. It's the blueprint for every living thing on the planet. Think of it as an instruction manual for how to build a piece of furniture, or in our case, a living organism, a living thing. So things like trees or insects, fish, people. And so the people part is the why do we care question and the answer to it. As our understanding of DNA has increased, we've seen improvements in medicine, forensic science, and a clearer understanding of how we all relate to one another, as well as how we relate to all organisms on the planet. For the what's it made of question, we're going to use the TI-Nspire file, DNA structure, to illustrate the structure and properties of DNA, deoxyribonucleic acid. Let's get started. Okay, so I have the DNA structure file opened in my TI-Nspire CX Premium Teacher software. You can use this same file in the student software also. This video is meant to focus in on the lesson about DNA structure. If you have the software in the file, you can follow along and pause the video and interact with the simulations. If you don't have the software, you should still be able to use this video to learn about DNA. That's what we're doing here. Okay, so I went to page 1.2. On page 1.2 it talks about DNA being part of all living organisms and the genetic instructions that make up an organism such as yourself. DNA is very simple and quite repetitive, and you'll see what that means in just a minute. But basically it's pretty neat. You have 4 building blocks that you use in various combinations to make all the structures in your body. It's a pretty cool thing to think about. Okay, page 1.3 goes over some instructions about the simulations that you'll interact with regarding DNA structure. So I'm going to jump over to page 1.4, and you can see the page numbers in the tabs above. And what I'll do is I'm going to click on these little clickers to illustrate the different parts of DNA, but let me explain what you're seeing on the screen. So what we have here is a picture of chromosomes, and a little circle or oval here is highlighting an inset of one of the chromosomes that's been condensed. And this might be, this is definitely a stylized representation of DNA just to get the point across of the different parts. And you'll notice there's sort of this gray helical structure, and then these very colorful rungs in between. So if you think of a ladder that's sitting against the side of a house or an apartment building, if you were to take that ladder and then twist it several times, it would look something like this. So the rungs of the ladder are here, and then the sides of the ladder are here, but just twisted up. Okay, so I clicked on the right arrow, and it says that the gray portion is the phosphate backbone. So a chemical molecule made up of phosphate that makes up this sort of framework for DNA. And then I click again, it says nucleotide. And basically what a nucleotide is it's made up of 3 parts. It's made up of the base itself, so one of the 4 letters that we'll talk about here in a few minutes, plus the sugar. And the sugar in DNA is the deoxyribose portion of the DNA name. So usually any molecule that has ose at the end of it, it refers to as sugar. In this case, it's deoxyribose. With RNA, which we'll learn about in another video, it's ribose. So DNA is deoxyribose, which means that it's missing an oxygen. Okay, so so the nucleotide is made up of the phosphate, it's made up of the sugar, and the base, all combined into one unit. So it's three parts in one unit. And then finally, the base pair. These are those colorful rungs of the ladder. So green and blue, red and yellow, and we'll find out what those different base pairs are. We'll learn the names of them here in a few minutes. On page 1.5, how many separate strands of DNA are in the double helix? Well, the name kind of gives it away here. The double helix is made up of two separate strands of DNA. All right, hence the name double helix. Next question is, the phosphate backbone is covalently bonded to A, a nucleotide base, B, a base pair, C, a double helix, or D, phosphate. The answer to this question is A, nucleotide base. So let's go back to that previous structure, and you can see that here. The phosphate backbone, that gray sides of the ladder, are connected to one base, one nitrogenous base. Now, when you bring the two sides of the ladder together, those bases connect, they bond, and form a base pair. But the answer to that specific question is the phosphate backbone is covalently bonded to a base pair. Okay. All right, the next section goes over Chargaff's rule, and we'll talk about what that means. Here's a little background. So DNA has 4 nucleotide building blocks. These are called bases. They're called adenine. We use the letter A to denote adenine. Thymine, which is T. Cytosine, the letter C. And guanine, the letter G. Before the structure of DNA was known, a man by the name of Erwin Chargaff, Austrian professor at Columbia University, made some observations about these bases that sort of helped us later figure out the double helical structure of DNA. So it wasn't until the 50s that we figured out how DNA is structured. Before that, there were a lot of people, a lot of scientists trying to figure out what it's made of. That wasn't that long ago, really. Next page talks about Chargaff's rules, that there's always the same percentage of adenine as there is thymine, and the same percentage of cytosine as there is guanine in any DNA sample. And so that was a sort of a clue for Chargaff that adenine and thymine were somehow related, and cytosine and guanine were somehow related. So, and we'll explore a little bit more of this in the next page with the simulation. But the next thing is the next observation was that the amounts of adenine and thymine, or cytosine and guanine, were similar on a single strand of DNA. Next page is page 2.3, and you'll see this little circle chart with A, T, C, and G. And this is to illustrate this point that some of the nucleo or the nitrogenous bases are related. So what you would do here is you would just click on this little clicker, and you'll notice as I change it, the amount of adenine and thymine change together. So when I have 26% adenine, I should always have 26% thymine. Also, for cytosine and guanine, whatever those numbers are should be the same always. Cytosine 22%, guanine 22%. Okay, and so that's a clue that those bases are related to one another somehow. So just to check, make sure you're understanding this. A couple of questions here based on Chargaff's rules. Which are the correct base pairs? So select all that apply. We discovered that adenine and thymine, and we'll go back here, we can see this, they have the same percentage no matter how much you start with. How much ever adenine you have that same amount of thymine. And again, same thing for cytosine and guanine. How much ever cytosine you have, that's how much guanine you're going to have also. So based on that, looks like there are two answers that are going to be correct. adenine and thymine, and cytosine and guanine. Okay, page 2.5 asks, if a DNA molecule is 29% guanine, what percent will be thymine? Now, at first glance, this question might be a little tricky. We just learned that guanine's not related to thymine directly. Guanine's related to cytosine, and adenine's related to thymine. So the question's asking you to think about, okay, well, I know I can only have 100%, and if I have 29% guanine, that must mean I have 29% cytosine. So let's do this. I'm going to open up a calculator screen. So I just clicked on a little button on my little calculator emulator, and the question asked if we had 29% guanine, how much thymine would we have? Well, to figure that out, we know that 29% plus another 29%, whoops, wrong button, there we go, plus another 29% is going to equal 58%. So that means that 29% guanine, and we know that cytosine has to be the same percentage, those combined equal 58%. So 58% of the DNA that we're talking about here in this special case, and it's going to differ from one bit of DNA to the next. 29 and 29 means it's 58% of your DNA molecules made up of cytosine and guanine. So now if we take, okay, 100% and we subtract that 58%, that means the remaining 42% of your DNA must be made up of adenine and thymine. Well, it only asks for thymine, so what we can do is we'll take 42% and divide that in half, and there's your answer. It should be 21%. So it's going to go over here, let's click on 21%, and we'll go ahead and move on to the next page. So hopefully that made sense on how I did that for you. And you can test yourself. You can, come up with some other scenarios. And this is a pretty common, assessment question that a biology teacher would use to make sure you understand the Chargaff's rule concept. Page 2.6, are there limits to the amount of any base pair in DNA? For example, can you have DNA that is 52% adenine? Explain. So, this is another question just to make sure you're kind of understanding. You can't have any more than 100%. And so if I had 52% adenine, that would also mean I have to have 52% thymine. Well, that's 104%, and we haven't even included the cytosine and the guanine. So all DNA contains those four bases: adenine, thymine, guanine, and cytosine. So this scenario is not plausible. We couldn't have 52% adenine. All right, let's see. So we're going to go to problem 3, page 3.1, and this is introducing a little more detail around what is a nucleotide, how does a nucleotide work, and this is kind of neat. And you can do this on the student software, teacher software, or the graphing calculator, TI-Nspire CX graphing calculator. If you mouse over to the little magnifying glass and wait till it changes to a little hand, see that? The open hand is there, but if I click the left mouse on my my keyboard here on my computer, or on a calculator, if you had to do it on the calculator, you would just you'd kind of move your mouse over, and then when you got to this point, you would just click down in the middle, and that'll grab that magnifying glass. And what we're going to do is we're going to move around the structure. Now, let me first explain what we're looking at. So what we see here, do you remember that gray part of the ladder, the two sides of the ladder? That's what this actually is. So these are phosphate groups chemically connected to a deoxyribose sugar, and you'll see these things repeat. And so phosphate connected to the deoxyribose sugar, and now I'm connected to a base, and another phosphate, another deoxyribose sugar, another base, and then the same thing on the other side. Now the 5 prime, that little symbol's prime, 5 prime and 3 prime ends refer to the number of the different number of carbons on those deoxyribose sugars. And so this is a 5-sided structure, and without getting into too much detail, the 3 prime and 5 prime refer to the 3 prime carbon and the 5 prime carbon on those deoxyribose sugars. Something else interesting to note, you'll notice that DNA, although it's complementary, you'll notice that the 5 prime starts up here and goes to 3 prime down there, but on the other end it's opposite. So 5 prime starts here and it goes to 3 prime. So these are parallel molecules, but they're called anti-parallel because they're going in opposite directions. And that's a really important concept when we get into DNA replication. Okay, but for now, let's just explore what it's made of. So if I take my magnifying glass and I mouse over these different parts, you'll see a little question mark appear. And when I pop up, let go of the mouse, and it'll pop up a little screen, and it says that for this one, we're talking about phosphate. Phosphate bonds with ribose to form a backbone that connects all the nucleotides within one strand of DNA. Okay. Alright, so let's grab that magnifier again and let's go exploring, see if I can find some more question marks to appear. Oh, there's one. Alright, so this is one of the 4 bases. This is cytosine. Cytosine always pairs with guanine, and it uses 3 hydrogen bonds. So this is important to know. This is one of those knowledge things that you just have to you remember, at least initially. But yeah, so there's 3 hydrogen bonds here that connect cytosine to guanine. So it connects here at this NH2 group at this nitrogen atom, and then again at this double bonded, this oxygen atom right here. And so I'm going to go ahead and jump over to guanine. I should see something similar. Alright, this base always pairs with cytosine by 3 hydrogen bonds. Same kind of deal here. So the guanine's slightly different than cytosine. That's why they have different names. But the same kind of thing here. I've got an NH group here, an oxygen there, and an NH2 group right here. But you'll notice 3 bonds. And if we move up to this green molecule, this is the adenine base. Always pairs with thymine, which we know from Chargaff's rule, and it has 2 hydrogen bonds. So here there's only 2. Now you remember cytosine and guanine require 3 hydrogen bonds. Adenine and thymine require only 2 hydrogen bonds. And so just to be thorough, we'll go to thymine. Always pairs with adenine with 2 hydrogen bonds. You'll see those bonds are here and here. Okay, so the thing we're missing, we haven't found yet, there it is the deoxyribose. This forms a bond with the phosphate backbone in one of the 4 nucleotides within a strand of DNA. So remember that deoxyribose means that it's a ribose sugar, but it's missing an oxygen. Okay, so that's why they call it deoxyribose. Alright, so that's a fun little slide, fun simulation you can explore, and just to get familiar with the different parts of DNA. Okay. Alright, so this next page is, does DNA have an overall charge? Explain. DNA does have an overall charge. That charge is negative, and it has to do, and I went back a page, it has to do with the phosphates. So the phosphates themselves carry a overall negative charge, which gives DNA that negative charge. And that becomes really useful when we start getting into concepts such as gel electrophoresis, which is basically the separation of DNA parts and pieces based on that negative charge. So we can split DNA up, we can cut it up into parts, and then the negativity of the phosphate group, I can apply a positive charge and get DNA to move through this thick gel, and I can separate it, and that's how forensic scientists are able to do DNA fingerprinting in crime scenes. Which structure is found in the backbone of DNA? There are actually two. We've talked a lot about this one here, phosphates, but part of that is also this deoxyribose. And let's take a look. Let's go back to that DNA structure that we talked about before. We got phosphate and deoxyribose. Phosphate and deoxyribose. We talked about this earlier. So actually that side of the ladder is made up of both the phosphate and the deoxyribose sugar with those nitrogenous bases in the middle. Okay. All right, so there are two correct answers for this question. Hydrogen bonds and base pairs are related. It is the hydrogen bonds that make the base pair. So you remember in that structure earlier, one more time, these are hydrogen bonds that connect adenine to thymine, these two. And these three bonds between cytosine and guanine, they're all hydrogen bonds. All right, page 3.5. Is the base pair AT bound more tightly than the base pair CG? Explain. Well, if you remember, when we looked at that structure, one of the base pairs has 3 hydrogen bonds and one only has 2. So, you know, you can reason that the cytosine and guanine have a stronger bond because of the 3 base pairs as opposed to 2 base pairs. All right, we're almost at the end of this lesson, and the next page is page 4.1. We've looked at the structure of DNA. We've talked about the phosphate sugar backbone, the deoxyribose sugar and phosphate that make up the backbone. We've talked about the nitrogen bases that when they're combined with hydrogen bonds, they form a base pair. We talked about what a nucleotide is, which the nucleotide is the phosphate, the deoxyribose sugar, as well as the base itself. Those 3 parts make up a nucleotide. All right, and now what we're going to do is we're going to play a little game where we can try to match these bases up. This is pretty obvious once you start playing with it, but it's still kind of fun just to mess around. And so you can click on these clickers and try different bases. You'll notice that cytosine is denoted by C, guanine G, thymine T, and adenine A. And it's pretty obvious how these things match up, but we can mess around and see. I'm going to get one wrong just to see what happens when I press the play button. It says, sorry, you have a base mispair. Well, that's not cool. Let me reset it and let's try again. You'll notice that the DNA structure changed, so you can change it to whatever, how many ever times you want to mess around with this, you can do. And this time, let's see if I got it right. Yay, nicely done. All bases are paired together, so I did a good job. And then finally, the last few questions talk about that pairing behavior. So both strands have the same sequence of DNA. Is this true or false? Well, let's go back and look. I don't think that's true. Let's see. Now they're complementary, but they certainly aren't the same. And so we can look and see this is thymine, adenine, thymine, cytosine, thymine. Well, that's different than this. This is adenine, thymine, adenine, guanine, adenine. So that answer is false. Both strands have the same sequence of DNA. That's not true. They have a complementary strand of DNA, but it's not the same. Next question on page 4.4, what strand of DNA can be used to solve the sequence of its pair? And that is true, because we only need one strand of DNA. We proved that earlier in this simulation. So I know that thymine matches up with adenine. We've shown that many times. And I know that adenine matches up with thymine, so I know that thymine must be the answer. And so on in that simulation. So that simulation is a lot of fun, just kind of illustrates how DNA matches up. Okay, well that's the end of this lesson. I encourage you to check out the next video, which will be covering DNA replication, and that should be posted soon. Thanks very much.